Yeah, so thanks very much for the introduction again and for the invitation to speak in this forum. So what I'm presenting today is a technology that we've been working on over the past 2 3 years, and it's based on previous technology, which is nano spray drying technology, and we have modified it to be applicable for the production and collection of nanoparticles with high yields. So it's focused on continuous production of small molecule drug nanoparticles, but it can be applicable for large molecules as well. So we use high pressure fluids, in particular supercritical CO2, to assist with the atomization of our drug nanoparticles. It's a work that we've been developing here at the center of at SSPC, but this particular technology has been most recently funded by one of the founding agencies in Ireland, which is the Enterprise Ireland, where they fund projects which are closer to commercialization rather than just platform fundamental projects there. So just kind of setting up the scenario there as a background, the focus that we've been trying to put on the need to produce in an efficient manner drug nanoparticles is due to the fact that new chemical entities coming out from the industrial pipelines have become more and more complex, and the molecular weights of those small molecule APIs have been increasing over time. And that means that those new chemical entities are getting more and more insoluble. So poor solubility is an issue, and that typically leads to poor drug efficacy, poor bioavailability. So we can see here in the graph, and this is data from a few years ago, but certainly it's quite actual and recent, and represents the chemical entities which are currently in the market, but also coming in the through the various clinical phases trials from the industry, which is between 70 up to 90% of new drugs are poorly soluble. And that doesn't mean that all of them will be poorly bioavailable, but it's very likely that it will cause issues in terms of bioavailability. So there are different ways or different methodologies that the industry looks at some stage before going into preclinical trials, try to look for ways to modify, to formulate their drugs in order to get the right dissolution rate, the right solubility, all of that. And one of the methods that has been increasing its popularity and has shown its efficacy, it's through the use of nanotechnology there. And in terms of nanotechnology, there are top-down methods and bottom-up methods. I don't want to go too much in detail on this, but just looking at top-down methods, which are seen here in the figure in the left, which is methods that the industry has been using very efficiently over the years, particularly the pharmaceutical industry. And that relates to breaking down drug particles using various top-down methods like milling and high-pressure homogenization, breaking down particles in a scalable fashion to produce micron-sized particles or even nano-sized particles. Now, these are energy-intensive methods, very efficient, very scalable, but they have their shortfalls there. And there's always a limit in terms of the minimum particle size that you can achieve using milling methods. Now, bottom-up methods are more advantageous in some ways because it can allow you to reach very, very small particles, because essentially you're precipitating or crystallizing your ABI particles from solution, from nothing, so that allows you to reach very, very small sizes. It has challenges as well, so those methods have not been implemented at the industry scale in the industry massively over there. But in terms of bottom-up methods, certainly liquid anti-solvent precipitation methods are methods that are used at small scale. They work very well, but scaling up is an issue there. And spray drying is very popular in the pharmaceutical industry. mostly for the production of microparticles, because when you go into the nanosize range, then you have an issue in terms of collecting those small particles there. Typically, you have electrostatic charge particles there. It's quite challenging. So over the years, we have been using supercritical CO2, mostly using coaxial nozzles to produce our micron-sized particles and nano-sized particles there. And one of the issues that we have been facing is trying to scale up those methods. So it works well at the small scale, lab scale, but collection yields are quite poor in terms of how much do you get after the drying chamber, after the cyclone, how much powder you get. And typically, production yields are quite small, like at the lab scale, 20%, 10%. But then as you try to scale up, typically, when you're doing scaling up of a spray drying process and you're producing microparticles, when you scale up, typically the yields get better. But when you're producing nanoparticles and you're trying to scale up, the yields get even worse, because nanoparticles, electrostatically charged nanoparticles, they get attracted to anything which is a surface. So if you have drying chambers of a spray drying device, the larger the drying chamber is, the more is the surface area available for the nanoparticles after spray drying to be deposited on the wall. So that means the yield get lower and lower and lower. So you reach easily 5% yield. And typically in the industry scale process, you are looking at 90% yield, 80% yield as a minimum there. So certainly that is an issue there. Now, supercritical CO2 technologies as a whole, we know that there has been a lot of interest. There was a lot of interest in the early 90s on different types of technologies using supercritical CO2, and then particularly with the pharmaceutical application there for pharmaceutical APIs. But then that interest kind of faded off, in the 2000s, and now it's getting back that interest again over there. And one of the issues that has been, I suppose, delaying the implementation of these type of technologies by the pharmaceutical industry is really scalability. Scalability, the fact that we're using high pressure, and when you're trying to scale up a process, even if it's just a high pressure vessel, an extraction process or a crystallization process using a high pressure vessel, when you're trying to scale up, then you're going to face certainly sometimes scalability issues, which happens to all type of technologies anyway. But then you're matching that with high pressure operation and safety aspects, and that's quite challenging. So scaling up high pressure vessels has always been a challenge, and the way we're trying to do is what can be done in a high pressure vessel. We try to scale down that into a nozzle which contains a small mixing chamber. So that's our high pressure vessel, and instead of scaling up a process when you're trying to produce microparticles in a high pressure vessel, and you want to scale up your process to get higher throughputs, we are actually scaling up a process while at the same time scaling down the size of our vessel down to a very small nozzle. And by doing so, we are not, I suppose, increasing the safety of our process. When you're scaling up a high pressure vessel, we are actually making it safer because we are scaling down our technology down to a nozzle. But a nozzle, which contains again a small high pressure vessel inside, but a nozzle that can be operating 24/7. So you can be spraying your particles, either as microparticles or nanoparticles over time there. So that thing on, rather than scaling up the size of your vessels, but scaling down into a nozzle will actually help to scale up the whole process, right? In terms of scaling up the throughput, but instead scaling down the size of your reactor there. That will allow you to make it continuous production of your microparticles or nanoparticles there. So again, we've been looking at using nano spray drying using high pressure CO2 for the production of microparticles and nanoparticles there. And it really started first when we're trying to crystallize our API, our API, which is previously dissolved in other organic solvent, and we can use polymers or excipients in case we want to do encapsulation, all of that. And These are typical high pressure processes that are used typically with high pressure vessels there. But again, when you're scaling up those type of high pressure vessels, it's an issue if you're going to scale up a process which is scaling up from 100 milliliters up to 10 liters or 100 liters, then you have an issue dealing with high pressure vessels there. So we do the opposite, which is scaling down the size of our vessel down to a coaxial nozzle, where here we have CO2 coming in, high pressure CO2, and our API solution being inserted there before CO2 comes in. So that means CO2 comes in, it saturates our solution and API precipitates. In this particular case, we do the same, but in a coaxial nozzle. So our superheat filled CO2 with the right pressure, the right temperature, mixes with our API solution continuously in this very small nozzle. And that in small nozzle, we can have nucleation happen, concentration happen in the nozzle, that's spraying afterwards in case we want to promote crystallization, or we can just having high pressure CO2 just acting as an atomizing fluid. So just applying pressure to the drug solution, just spraying through the nozzle over there. And like doing that, we can then control the size of our particles from the micro size range down to the nano size range, at the same time as controlling other particle attributes like polymorphic form, solid state form. So that's converting a batch process into a continuous process, but actually scaling down the size of our high pressure vessel down to a high pressure nozzle there. That means allows us to do the drying of our spray dry nanoparticles at their atmospheric pressure, 1 bar, while restricting the high pressure only into the nozzle itself. So all the rest will be under 1 bar, and the high pressure is just located at a very small scale within the high pressure vessel there. So that's nano spray drying or micron size spray drying using high pressure CO2. And this is just a view of one of the oldest high pressure spray dryers that we have, where we have the coaxial nozzle, and beforehand we have small mixing chamber where we mix the CO2 and the API solution. So we can achieve supersaturation conditions inside the nozzle, or we can be below the saturation point in case we just want to mix the two fluids, so API solution and the CO2, and spray it afterwards. Controlling the size of the nozzle and controlling the solution flow rate, CO2 flow rate, all of that will allow us to control droplet size, and the droplet size will ultimately impact the particle size that we are producing here. Obviously, when we are producing crystalline particles, droplet size does not completely control particle size, because crystallization will be difficult to control particle size in the crystallization process. We can control particle size, but it is sometimes it depends on the API we are working on. In some cases, we might want to achieve 200 nanometers particle size, but with some APIs and with some polymorphic forms of those APIs, we might get 300 or 400. Just depends on the type of API that we are working on. But again, this allows us to produce different types of particles, so small molecule APIs and certainly some large molecule model systems over there. So one of the first APIs we were working on was carbamazepine, which is a small molecule API, but it's quite polymorphic. So there are 7 reported forms of polymorphic forms of carbamazepine. And by just using nanospray drying, by controlling what's happening in the nozzle, so that means controlling the temperature, controlling the pressure, and the CO2 and the solution flow ratios within the nozzle, we can achieve saturation conditions in the nozzle that allows us to nucleate API that we want before actually crystallization happens. So that means crystallization will happen after the nozzle, but we can template the polymorphic form that we want in a very short residence time, where the CO2 mixes with the solution, or the API solution inside the nozzle. For just a few milliseconds, we can induce the formation of a particular polymorphic form here in the nozzle, and then try to promote the growth of that polymorphic form after the nozzle as exits within the dry chamber there. So for carbamazepine, we have publications with other APIs as well, but with this API, we published a paper a few years ago where we've shown that we can depending on the conditions that happen in the nozzle, but certainly in the drying chamber as well, we can get amorphous form of kermesbean, or if we induce the nucleation of kermesbean inside the coaxial nozzle, we can produce metastable forms of kermesbean form 1, form 2, or producing the stable form of form 3 by controlling temperature, pressure, solution, and CO2 flow rates, but also adding small proportions of additives into our API solution before the CO2 nanospray drying process there. So that's nanospray drying. It works very well. We can control particle size, solid state form, amorphous versus crystalline. And if we are producing crystalline material, we can tweak the polymorphic form that we are producing. The challenge was always collecting the API particles. So it doesn't matter how well you control your process, how well you control the attributes of your particles, if you only collect 5% of what you have actually sprayed. You want to go from 5% up to 80% and 90%, because most of the nanoparticles, they get trapped inside the walls of the drying chamber. They are so electrostatic because they're very small, they're charged, so that they coat your drying chamber and not much gets out through your collecting vessel there. So based on that, we have developed further the nanospray drying technology into the CR nanotechnology, or two different types of CR nanotechnologies. So it's a nanospray drying process, but it's actually a nanospray coating process. And in this particular technology there, which we took our nanospray drying chamber and we have slightly modified it in a way that we are collecting our nanoparticles, coating those onto the surface of excipient particles there. So just going through it in more detail. So this is our spray drying process, very similar to this one over here. And the difference is when we are mixing supercritical CO2 and API solution, that is always the same. We can control the nucleation if we want to proceed, if we want to promote the nucleation of our API before the droplets of the solution are formed, before the nozzle exits. We can control the nucleation, and so applying the CO2 with dissolvent effect. CO2, high pressure CO2 typically has very good miscibility with most organic solvents there. And it doesn't have good solubility with many APIs. So that means that your API, which is dissolved in a particular organic solvent, as it goes through here, it mixes with the supercritical CO2. Supercritical CO2 will supersaturate your solution. That means that your solvent from here will have affinity to mix with the CO2, but API won't, so the API will precipitate out as particles there. To avoid precipitation, we have a small mixing chamber, so you don't have much time for the solution and the CO2 to mix to precipitate API particles. What happens is we template the formation of the nucleus of a particular API polymorphic form, and then immediately it's pressurized and sprays to the nozzle. So this is a continuous process. These fluids are continuously going in and going out. So you don't have much time here. The residence time is about a few milliseconds there. So we can control nucleation still as our nanospray drying process. But then as the droplets are being formed and being dried, those nanoparticles which are being formed are being coated onto excipient particles here, which are here on the top of this mesh. So we place a mesh, a stainless steel mesh, with certain openings here to allow for the gas to pass through. So here we have high pressure CO2, supercritical CO2, and here, because it's 1 bar, CO2 is a gas, it's a vapor. So CO2 and the solvent vapor will pass through these holes over there. But the excipient particles which are sitting here on the top of this mesh, as the CO2 flow comes in, these excipient particles there, they start fluidizing and moving around, and they get the API nanoparticles to coat on the surface of the excipient particles there. So what's happening is we have a nanospray drying process, but then it's more a spray coating process because you're producing your drug nanoparticles, and your drug nanoparticles are being coated onto the surface of those excipient particles. So we can look at any size of nanosized particles here as low as 100 nanometers and being coated on the surface of 50 microns, 100 microns, 200 microns, any size you want for your excipient particles there. So you have if you want, you have control nucleation in the nozzle, you have control atomization and spray drying in the drying chamber, and then you have isolation of your drug nanoparticles onto micron size carrier particles there. And then afterwards, you're discharging your formulation here through here. So then you have a micron size formulation because you have micron size excipient particles, but those excipient particles will have drug nanoparticles coated on the surface. So that means that your nanoparticles now, they still have high dissolution rate and high apparent stability because they are drug nanosized particles. But your formulation will behave as a micron size formulation because your excipient particles will have good flowability, good biological properties, and can be directly compressible into a tablet, which if you are dealing with just drug nanoparticles alone, first, the collection yields will be very low because your drug nanoparticles will be coated on the walls of your drying chamber, and you'll not be collecting enough particles to do any formulation. And then even if you were able to collect enough material from your drug nanoparticles, the rheological properties will be very, very poor because you're dealing with static particles and very small particles. So you blending these drug nanoparticles with other excipients to make your tablet will be very, very challenging. Well, over here, we have another formulation which can be directly compressed into a tablet. So in one step, in one process, you are bringing your API solution going through precipitation or crystallization if you have to produce crystalline particles and not amorphous. But you can also produce amorphous if you want. And then going down to producing a drug product intermediate, which can be directly being converted into a final drug product. So this material, this product can be going directly into a tablet machine there. So that's our semi-continuous process. Semi-continuous because we are adding our excipient particles here. We have to wait while we are spraying the drug nanoparticles until our excipient particles are coated, and then we discharge these particles down and we reinitiate the process again. This is our batch process, and we've also fully converted our batch process into a continuous process there. And the methodology is the same, but the way the technology works is slightly different. Because here in the batch or the semi-continuous version of this technology, we have a high pressure nozzle, coaxial nozzle, where the CO2 and the API solution mix, and then you have a spray into atmospheric pressure drying chamber, and you have your excipient particles here. Here is different. So what happens here is you don't have one nozzle, you have several nozzles on the walls of your drying chamber. And we can use as many as we want. We only use 4 or 6 nozzles, but we can use many nozzles there. And in each of those nozzles, if they are being used, you have your CO2 and your API solution being mixed in the coaxial nozzle and being sprayed. And what you have on the top is actually the excipient particles. Your micron size granules or excipient particles there are being dropped through this hopper, and then they are falling down by gravity here. As they are falling down, they are being coated from different directions by drug nanoparticles, which are being sprayed through these nozzles over there. So just looking into a bit more, zooming in, we have these nozzles spraying drug nanoparticles over here, while your excipient particles, micron-sized excipient particles, are falling down and are being coated by drug nanoparticles. And they are continuously leaving your drying chamber to the sidelong, into a collecting basket there. So in one continuous process, the excipient particles are going in, and they come out fully coated with your drug nanoparticles, achieving very high collection yields, as I'm going to show afterwards. So this is a fully continuous production and isolation process, converting API solutions into a drug product intermediates, which can be directly compressed into a tablet. So in terms of the process itself, it can telescope through multiple unit operations in a manufacturing plant. So in some cases, when you're using a crystallization process and you need to remove your solvent after crystallization process, you need to filter, you need to dry it, you need to mill it to control particle size that you're going to get. Since crystallization typically leads to large crystals, You want to break down the crystals down to a size that you need, and then you need to go through different units, operations, processing steps until you reach your finished product. With this type of technology, it can eliminate the need from any steps there. So you can produce nano-sized particles in a micron-sized formulation directly in one single step, and that allows you to reach the finished product much quicker. And the principle here is always, you need to use excipients, yes, but you can fully coat your excipient particles by your drug nanoparticles there. So making use of the full potential of your drug nanoparticles in terms of high dissolution rates and higher aberrant stabilities, but certainly having excellent rheological properties that allow you to go through downstream processing down to your finished product there. Either the finished product is a tablet or it's a capsule, or even if it's a product for inhalation, which is something that we are working on that as well. So just going through in terms of some results there, looking to the type to see how much time I have available. Just giving you some examples. Now we have been using different types of ABI model systems for our nano spray drying process, which uses high pressure CO2. And then also with our CM nanotechnology, where we do production of nanoparticles and coating those nanoparticles into micron-sized excipient particles. So we have been using BCS class 2 API model systems like the ones we are looking at here. And then also BCS class 4 as well. And now we are working with other types of APIs which belong to these classes. So BCS class 2, BCS class 4, they all have poor solubility over there. And with some examples, for instance, for BCS class 2, like carbamazepine, risperidone, and ketoprofen, which have relatively poor solubility in water and poor dissolution rates. We process each of these APIs using our CF nanotechnology. So we were spray drying or nano spray drying each of these APIs and coating onto the surface of excipient particles there. And the example here we are showing is we have used MCC, which is microcrystalline cellulose, D50 of 100 micron size. And our MCC is being placed here on the top of this mesh in case we are using our semi-continuous CF nanotechnology. So the MCC, on the surface of MCC, we have our ABI nanoparticles being deposited, or the MCC is actually being put on the hopper and going down through our CF nano continuous prototype and being fully coated with ABI nanoparticles there. So what we are seeing here in the SEM images is our large micron-size MCC particles, which are here on the background. And on the surface, we have the position of our ABI drug nanoparticles there. So we have here small loadings. In this particular example, we have 10% loading. We have 20% loading of our ABI nanoparticles, and we can achieve up to 40% loading efficiently of our ABI nanoparticles onto the MCC. Here is carbamazepine coated onto MCC particles. Here we have risperidone, and here we have ketoprofen here being coated onto MCC particles there. In terms of collection yields, so we went from 5% collection yields in our nano spray drying process, where we didn't have any excipient there. We were just nano spray drying our APIs there and getting low collection yields. We went from 5%, 10% up to 80% and above that using each of these API model systems there. So controlling the particle size, the average particle size down to 100, 150 nanometers there, and achieving very high yields of our drug nanoparticles. So that's the yield that you collect in your collecting vessel. That's our API coated on the micron-sized excipient particles there, as well as allowing us to control the polymorphic form of our API there. So small particle sizes, ability to control the solid state form, amorphous or crystalline, and if it's crystalline, controlling the polymorphic form of our crystalline particles there. Now comparing this, our CF nanotechnology, so nano spray drying with our CF nanotechnology, and comparing this with the traditional spray dryer or nano spray dryer, we can achieve much higher collection yields there. That also allows us to scale up our process into a pilot scale version. which is the case for our continuous technology there that we are scaling up into continuous pilot scale version there. And currently that allows us to maintain high collection yields and high production yields of our drug nanoparticles there. So just giving some examples before finishing the presentation in terms of the rheological properties of our nanoformulations, our API nanoparticles coated onto the micron size cellulose excipient particles there. Here we have CBZ carmazepine, our API coated onto MCC. Here we have risperidone or ketoprofen coated onto our micron size cellulose particles there. That's the compressibility as a function of the applied normal stress that allows us to see if our formulation has a good compressibility, a good flowability there. So if we compare with MCC, MCC is known, microcrystalline cellulose is known to have very good rheological properties, very good flowability, very good compressibility as well. So we are looking at the blue squares there. As you increase the applied normal stress, your compressibility increases in a way that you are in a good region in terms of compressibility. We don't have here data for our spray-dried API nanoparticles alone. If we added, this will be very, very hard. We'll go into a very poor region in terms of compressibility. Now our API nanoparticles, since they are coated onto MCC, we see good compressibility properties for our API nanoparticles. Each of these types of API nanoparticles coated onto MCC, they have more or less similar behavior in comparison to the excipient alone there. So that's for 10% loading of API onto MCC, and this is for 20% loading there. This is solution rate as well. So we look here into these API model systems, these BCS class 2 model systems there. We have risperidone as received, carbamazepine for 3, ketoprofen as received as well. Particularly risperidone, this solution rate in water is relatively poor over time. So you reach low percentage of the solutions. But our API, if it's formulated with our CM nanotechnology, if we are producing nanoparticles coated onto the MCC particles, the dissolution rate is much higher for each of those APIs there. So we are looking at 10% loading of our API nanoparticles coated onto MCC particles. And then for 20% loading, also very good dissolution rate. So our drug nanoparticles, they behave as nanoparticles, although they are part of a micron size formulation there. This is mostly it. So just showing that we're using high pressure CO2, it allows us to control, as we all know, solid state particle size as typical with other atomization based CO2 technologies there. But in our case, we can achieve very high collection yields, high throughputs, and allows us to scale up the size of our drying chambers while still maintaining high collection yields of our nanoparticles there. So it allows us to work in the batch operations, semi-continuous, or even a full continuous operation to produce our nanoparticles there. That's it for my presentation. This is just a snapshot, a picture of our Bernal Institute, which is here at the University of Limerick, where the SSPC Center, Research Center is part of, where we have SSPC is a research center which combines 9 universities or 9 research performing organizations in Ireland and 20 pharmaceutical industry partners which are part of the SSPC, and they fully fund the center here. This is just the website for my group and the website for SSPC. And this is my email in case you want to reach out for any questions or comments that you'd like to have. That's it. Thank you very much for your attention on this talk. I'm happy to answer any questions you might have.
Thank you for your talk. I have a small question on the solvent choice of your API. So you said you use organic solvents that has to be soluble in supercritical CO2. But you also showed some nanospray dried BSA and lysozyme, which are enzymes and proteins, which do not like those organic solvents. So how do you do nanospray drying of those particles or those proteins, and which are
Sure. No, we did nanospray drying with it using high pressure CO2. In some cases, we have used nitrogen. Just going back to the slides there. So this is for our albumin BSA. We have used water only. There's only water. So that means we have used high pressure CO2. In some cases, we have used nitrogen, but there was no mixing of this CO2 with the solution, because high pressure CO2 is not really soluble in water. So high pressure CO2 just was used as an atomizing agent, as an atomization enhancer. So there was no nucleation. There was no saturation of our aqueous solution inside the nozzle under high pressure. This was just aqueous solution was mixed with the high pressure CO2, but it wasn't, the water wasn't dissolved in the CO2, so there was no supersaturation event, there was no nucleation, it was just spraying through it. So we can still produce nano-sized particles, micro-sized particles. We need to dry efficiently the water. Now that means we cannot increase much the outlet temperature in order to avoid the naturation of our protein there. We have to use other things, which is we have to use additional inlet of a drying gas like nitrogen or hot air over there. But in this particular case, when you're using fully aqueous solution, you're not nucleating anything, which we don't need it. The nucleation or the anti-solvent effect of CO2 is used when you use organic solvents, where the organic solvents will mix and dissolve with the CO2, and then the API will precipitate out. So this is just nano atomization there. With lysozyme, we have used lysozyme as here, just using aqueous solutions there. And here we have used lysozyme with up to actually 70% of ethanol there. And in this case, we see formation of fibers, and we see some loss of activity, not much. So here we have 100% activity, more or less, and here we have 70% of activity. But lysozyme is a very, very robust enzyme, very robust protein, and it does not fully represent what we will get if we were spray drying or nanospray drying monoclonal antibodies or very sensitive proteins there. But yeah, if you want to have nucleation or controlling inducing supersaturation, then you need to use a solvent which is mixable and has good stability, mixability with CO2, which is not the case for water.
Do we have any more questions? No more. Oh, another question. Ruben, there we go. And sorry, again, another question. Have you ever tested your methodology against a fluid bed coating of those MCC particles? So you have then continuous layer of your API. So is it better if you have a nano particles on your MCC than a continuous layer?
So not sure if I understood correctly the question. So you're asking if we have used fluidization of our tech? No,
a fluid bed coating of your particles. So you have then a continuous layer of your API. So you have the effect the same thing, but instead of you spray just a solution of your API onto your particles, and you dry with temperature and with an airflow. So you have a continuous layer of your have a small layer.
Yeah. So I suppose each of these technologies there will have advantages and disadvantages there. The disadvantage here when we're using this semi-continuous process is that there's only so much you can put here on the top of this mesh in terms of the amount of excipients particles. So that typically is 20 grams, 30 grams in the size that we're using for our dry chamber. And that allows you to, while you're coating your nanoparticles onto the excipient particles until everything is mostly coated, and then you discharge and you insert more particles there. So you cannot put here 1 kilogram, right? So there's only a certain amount that you can put there. While over here, you can add significant amounts of excipient particles continuously and get in very high yields. Now, in the beginning, when we were developing this technology, it was that easy. We had to optimize nozzle, number of nozzles, nozzle direction, all of that. But this is more efficient. In terms of throughput, we can reach up to 100, 150 grams of excipient particles per hour going to our chamber there. We're trying to scale up the process there. But this allows us, when you are scaling up, it allows us to continuously, so far what we did so far is getting the same throughput and getting the same efficiency and the same type of particles that we're producing. The challenge here is we have this kind of a small scale, it's a laboratory scale spray dryer drying chamber. If we scale up so far, and we are still working on this, if we scale up the size of this drying chamber to a much larger scale, like almost a pallet scale, the efficiency of the drug coating, it goes lower. So that means the distance between the nozzle and the place where you have your excipient particles, it's critical. Because if this is too close to the nozzle, you get your excipient particles wet. So your atomization of your solution does not occur efficiently. So your droplets do not dry efficiently and they splash against your excipient particles there. If this is too far away from your nozzle, what you get is fully dried drug nanoparticles when they reach your excipient particles there, and that coating is not efficient. And you lose a lot of drug nanoparticles on the walls on your drying chamber. That means the larger this is, the size of your drying chamber, the more surface area you have in terms of the walls, and the more your drug nanoparticles will go to the walls if they are not trapped by the excipient particles there. So as soon as the drug nanoparticles are coated onto the surface of those excipient particles, that's it. You've got your nanoparticles there on the surface. But if this is too far away, that means that you have much more surface area of your walls for your drug nanoparticles to be deposited on the walls before they actually reach the excipient particles there. And that's the issue we are facing in terms of scalability of this semi-continuous process. So it's ongoing, but so far we have seen those challenges. As we try to scale up, yields get lower and lower and lower. Because distance from here, the nozzle, and the place where you have your excipient particles there is also very critical there. And here we can optimize the distance between the nozzle and the area where the excipient particles are. So this distance here, we can optimize by working with the diameter of your chamber. And we can make the chamber as tall as we need. So if we need more throughput, we increase the height of your chamber, so you have more residence time for particles to go through and to get codes over there. So there are advantages and disadvantages. The advantage here is you can fully adapt this to a conventional spray dryer just by modifying the nozzle to have high pressure CO2 there, and to put a mesh there in a lab scale spray dryer. This is more difficult to adapt to a conventional spray dryer. If you take a conventional spray dryer, you need to make holes to accommodate different nozzles, so it's doable, but it's more difficult. While this is, in theory, you can take a conventional spray dryer and just do small modifications. There you have your semi-continuous CM nanotechnology there. So it's a balance between advantages and easiness of adaptability to conventional spray dryers.